Hello, today we are diving into transcription and translation. So we're going to kind of dive into the idea of protein synthesis. This particular set of notes is the basics. This is stuff that you should have learned in year one. And so this should hopefully, fingers crossed, be like a review for you. But if you don't remember what happened in year one, that's fine. That's why we have this set of notes. But hopefully some of this is just sort of ringing bells for you and it's not all brand new information. So let's get started. What is the purpose of this? So transcription and translation produces genes, right? You're reading DNA, you're creating amino acids, polypeptide chains, and eventually you're creating genes. And these genes are instructions for proteins that the cell needs to use to carry out its everyday functions. So genes are the instructions for proteins, and proteins do what we need to do all day, every day to be like a functioning human being. In transcription, the gene is converted into mRNA, which is then posted to ribosomes. And then the ribosomes, which is like translation, translate that message into a polypeptide. And the sequence of amino acids is determined by the gene. So the gene are the instructions, like that's key here. The gene determines the sequence of amino acids, which then determines the properties of the finished protein. It all starts with the gene, though. The central dogma of genetics is sort of this idea that we were just talking about, right? The gene then transcribes into the mRNA, which is the message, which then translates into the polypeptide or the protein or the amino acids, however you want to say it. They all are sort of interchangeable terms, but you start with the gene, it transcribes into the message, and it ends up translating into the product. So the genes are codes for making polypeptides. The DNA is stored in the nucleus, and it cannot leave. DNA cannot ever leave the nucleus, but the polypeptide is produced in the cytoplasm by ribosomes. So the only way to get the gene into the polypeptide is with the use of mRNA, because it can travel from the nucleus into the cytoplasm and take that gene's message and make the amino acid. And again, the sequence of bases just tells the ribosome which amino acids to use. When we're talking about DNA and RNA, which we have been this whole time, I just want to reiterate a few key differences between them. In DNA, you have the sugar deoxyribose. In RNA, you have the sugar ribose, so different sugars. DNA is a double helix, RNA is a single helix. And the nucleotides that make up DNA are cytosine, guanine, adenine, and thymine, whereas in RNA you have cytosine, guanine, and adenine. Those are all the same, but instead of thymine you now have uracil. Uracil is complementary with adenine, just like thymine is, but in RNA, thymine does not exist. So T's do not exist in RNA. So the basic definitions of transcription and translation fall under the umbrella term of protein synthesis. So transcription is when RNA polymerase, which is an enzyme, RNA polymerase makes an mRNA molecule that is complementary to the DNA. So if the DNA said like ACTG, then the RNA would be UGAC. I think I remembered that correctly. But A goes with U, T goes with A, and C and G go with each other. Transcription happens in the nucleus because remember, DNA cannot leave the nucleus, which means that if we're doing anything with the DNA, like if we're trying to make complementary base pairs, if we're doing DNA replication, anything with DNA happens inside the nucleus. Whereas translation happens outside of the nucleus. So the mRNA is sent out of the nucleus and attaches to ribosomes. Ribosomes use complementary base pairing again, so we're always kind of using this complementary base pairing, but ribosomes connect mRNA to tRNA, so message RNA to transfer RNA, and both types of RNA use the same nucleotides, right? A, C, G, and U. And so when you are base pairing at the ribosome, you are base pairing the mRNA with the anticodons, which are connected to the amino acids. And I'm going to show you a picture of that in a second. See, it was only a second. So this right here is a tRNA. So this tRNA at one end has an anticodon, and at the other end has the specific amino acid that they can carry. Every tRNA molecule only carries one specific amino acid. And so the way that this all works is the mRNA will like hook up with a ribosome, and it will have its complementary DNA, right? So this mRNA is the complementary base pairs from the original DNA that it transcribed in the nucleus. And so these letters are the complementary base pairs, and they will match up complementarily, if that's a word, with the tRNA. So see, AUG is a start codon. It is always methionine. AUG, and then the tRNA has the complementary nucleotides, right? A goes to U, U goes to A, G goes to C, and that tRNA is carrying methionine, or met. And then the next one is just dependent on whatever the nucleotides are. So AAA would match up with UUU, and this would bring lysine. Okay, and then you would just continue to read it. This mRNA would continue to move through the ribosome, and the tRNA would continue to come up and match up complementarily with the mRNA and release the amino acids. I've been saying some of these words, and I want to make sure that we're all sort of on the same page here. Like when we're talking about DNA and amino acids and complementary base pairs, we're talking in terms of triplets. Those triplets are called codons. So when you transcribe DNA, you transcribe it in triplets. You transcribe it into codons, right? You create mRNA codons, and those codons make up the genetic code. So each gene will start with a start codon, that is methionine, that is that MET, that's AUG, what we just saw in the last slide. And all will finish with a stop codon at the end. There are specific letters that code for a start codon and specific letters that code for the stop codons at the end. They don't change. That's just always what happens, okay? There are 64 different combinations of amino acids, right? You can use different letters to get different amino acids, right? But infinite polypeptides, right? Because you can have as many amino acids as you would like, and you can put them in any order. So the type of protein you can create is infinite, but there are only 64 combinations of letters that will make up amino acids. Okay, and just as a reminder, the genetic code only has 4 letters, right? A, C, T, and G. That's the 4 letters. So again, DNA triplets, that's a triplet, right? Every 3 letters is a triplet. That's a codon. That codes for the mRNA codons, and those will match up with the anticodons on the tRNA, which will bring in these polypeptides. There are 64 possible combinations here, but there's only 20 amino acids. So although these letters there can be 64 possible combinations of these letters, you're only making 20 amino acids. So what does that mean? What does that mean? It means that it's degenerate. So I'm gonna skip that first bullet point for a second. I'm gonna talk about the fact that it's degenerate. And what does that word mean? Because you've maybe heard this in another context. It basically means that these codons are interchangeable, right? There's 64 codons, but there's only 20 amino acids, which means that the same amino acid can be coded for by more than one codon. That means different letters can code for the same amino acid, right? Because there's only 20 and there's 64 letter combinations, so multiple letter combinations can code for the same amino acid. And that's a good thing, because that means that even if there's like a base substitution or there's a mutation or something that we're not accounting for, the chance of a negative effect is a little bit lower, just because the same amino acid could be produced even if the letters change, right? So the term degenerate in relation to biology is sort of just means that it's interchangeable. In addition to that, the genetic code is universal, right? All living things use the same code, all living things have the same bases, and all codons code for the same amino acid no matter the organism. This is why you can say that, like, oh, humans share X amount of DNA with like a banana or a chicken or a shark, whatever it may be, because we're all using the same letters. We're all using the same codons, we're all using the same proteins. It all means the same. It's just the order in which these letters are found. It's pretty incredible, actually. So, this is, I just like, I'm giving you lots of visuals so that hopefully you're like remembering all this stuff. So, in this example, DNA, this has 21 nucleotides. If I have 21 nucleotides, that means I have 7 codons, right? I have 7 triplets. And if I have 7 triplets, that means I have 7 amino acids. Oh wait, no, we don't count the stop codon as an amino acid. I mean, yes, technically it is an amino acid, but because it is just coding for stop, we don't really consider it part of the polypeptide. So yes, there are 7 codons. Yes, there are 7. You would find out what these 7 amino acids are, but once you realize the last one was stop, you would like subtract it from your total. So in this example, again, met would be first, AUG, that's our first one, right? We're only focusing in where this line is, or where these colors are. AUG, met, that's our first one, and then we go through, we see the other ones, and then we see this last one, UGA, that codes for stop. So the number of amino acids equals the codons minus 1, right? You don't count the stop, that's what I just said. And so there are 20 amino acids and 3 stop codons that are coded for in the like translation tables that you could use to determine what these amino acids are. So let's review. In transcription, you start with DNA. In translation, you start with mRNA. In transcription, the genetic code is in the form of triplets, which are sometimes called codons anyways, so that word is like sort of interchangeable. In translation, it's only called codons. The output of transcription is the mRNA, which then goes into translation, but the output of translation is the polypeptide or the protein. Transcription happens in the nucleus, translation happens at the ribosomes, the rough endoplasmic reticulum / the cytoplasm in the ribosome. So, and then the molecules used, you have in transcription RNA polymerase and RNA nucleotides, that is an enzyme, and then the nucleotides that make up mRNA. And then in translation, you're using the ribosomes, tRNA, right, transfer RNA, and then amino acids. And then just to sort of finish us off, how are amino acids joined together? It's called a condensation reaction. So when 2 amino acids line up near each other, you do not need to know the actual chemical structure of amino acids. Oh my god, that would be crazy. That would be so much for you guys to know, but you don't need to know that. But when amino acids are next to each other, you see that you have an OH and an H right next to each other. Those will combine and turn into water, and so that water is removed and the peptide bond is then formed between the carbon and the nitrogen here. And so once water is removed, those elements will bond with each other, forming a peptide bond, and water is a waste product, and so it will, for every amino acid, it will go through this condensation reaction to create that peptide bond between them and connect them. And then last but not least, the one gene, one polypeptide hypothesis. So we love to talk about scientists. You're going to need to know a ton of scientists for this unit. So this is one of them. So at first, and when I say at first, I mean like 60 plus years ago, it was thought that one gene equaled one enzyme. Like that was it. There was no other information that could be given from that one gene. It was always one gene, one enzyme. And then it was modified to one gene, one polypeptide. So one gene would always create the same set of amino acids in a row, which is a little bit more accurate, right? We don't always make an enzyme, but we do make one gene will always code for the same set of polypeptides. And this was done by Beadle and Tatum, which are just two scientists that figured this out. But as we continue to learn more about it, of course, we have exceptions, right? Because we have different versions of genes, right? We have brown eyes and blue eyes, and we have, attached earlobes and unattached earlobes. So all these different alleles, it's still the same gene. It's still the same, beginning part of your genetics, but the alleles code for different things. So how are we getting these different alleles if we're under the assumption that one gene equals one polypeptide? Of course, science is never done. We will continue to explore this question. And as research has continued over the years, we're getting more and more exceptions, which is just the nature of science. So some genes control the expression of others, and we talked about this in a previous unit where we were talking about fur color in guinea pigs, whereas like if you had one gene, you could either express another color or you would become albino because you couldn't express any pigment, right? So some genes control others, which means that no polypeptide would be produced in that situation. And sometimes some genes code for RNA, which just doesn't produce a polypeptide at all, right? There's no start codon, so we don't know the function of what that gene is actually coding for. Like when it does code for that RNA, we don't know what it's then doing. We're working on that as scientists. The community is continuously studying this. We are trying to figure out what all of our DNA does. We are continuing to learn that what was once thought of as junk DNA is not actually junk DNA. And so these ideas, these concepts will be continuously fleshed out until we fully understand what is happening with our DNA. But until then, we're going to go with what we have sort of seen, and so we're going to go with the one gene, one polypeptide hypothesis with the understanding that there are exceptions to this rule. All right, great job. I'll see you next time.